turbo engine running.
Absolute genius.
Get this.
Welcome.
This is the show where we bring you
Science.
What that essentially means is
discoveries,
Advances.
Questions. Research.
technology. Unbelievable.
Without further ado,
This is The Naked Scientists. Hello, welcome to The Naked Scientists, the show where we bring you the latest breakthroughs in science, technology and medicine with me, Chris Smith. Coming up, body scans give us new insights into long COVID. Also, scientists discover the switch that triggers a mosquito's bloodlust, but can we turn it off? And we'll take you on a whistle-stop tour of the Royal Society's Summer Science Exhibition.
From Cambridge University's Institute of Continuing Education, this is The Naked Scientists.
First this week, scientists in the United States have been scanning patients with long COVID symptoms in a bid to assess the long-term impact of the SARS-CoV-2 virus. They've done it using a special imaging technique that lights up when it sees inflammation. Michael Peluso is at the University of California, San Francisco.
Well, the purpose of this study was really trying to understand what's going on with the immune system on a total body level after a person has COVID, and whether that could be related to long COVID.
How?
So we know from studies of blood tests that people after they have COVID have sort of lingering inflammation, and that people with long COVID may have more inflammation than other people. Their immune systems may be revved up for quite some time. But the blood is only a small window into what's going on in the totality of somebody's body. And so what we did in this study was try to understand what the immune system was doing in all of the various tissues, basically from head to toe in a person in the months or even years after they have COVID.
And how can you actually ask that question in a living person without taking them to pieces, obviously? How do you know what the activity of the immune system is on a tissue by tissue, organ by organ level?
So a few years ago, before the pandemic, my colleagues at UCSF, Kim Henrich and Henry Van Brocklin, actually developed this special PET scan with what we call a tracer, basically a molecule that we inject into people's veins, into their arm, and it goes throughout their body and basically lights up tissues that have increased levels of immune system cells trafficking into them. People do this special scan, and the areas of their body that are particularly active with certain types of immune cells called T cells light up on the scan compared to tissues where there aren't a lot of those types of cells. And so what we're basically able to do is look tissue by tissue, region by region, at the level of immune activity in each of these different places. So in the brain, in the lymph nodes, in the spinal cord, in the heart, in the lungs, in the liver, in the GI tract. And that gives us a sense of where that signal of inflammation that we might be seeing in blood is actually coming from. It has to be coming from somewhere. And so this was sort of a look into where it could be coming from.
Just in people with COVID or a range of patients?
So the purpose of this study was to really understand what happens to people after they have COVID. And so the majority of the patients that we scanned were, 1 month, 2 months, 3 months, 6 months, a year, 2 years after COVID. And many of the participants had, what we call long COVID, symptoms that persist and are otherwise unexplained after a person has COVID. But something that was really important that we did in this study was, because our program existed before the pandemic, we were able to use archive scans from before anybody on the planet had COVID to understand what was the normal level of immune activation, 5 or 10 years ago. And what we found basically across the board was that in people who had COVID, their tissues lit up compared to what was typical before COVID existed.
Did you look at people who had and hadn't been vaccinated against COVID? Because could that also be playing a role?
We typically record when everybody in the study gets their vaccines, and we did not actually scan people in proximity to getting a COVID vaccine. So the participants in the study had to be several months away from having gotten their most recent vaccine. And a number of participants in the study participated, before vaccines even became available. So we don't think that the findings are in any way attributable to vaccination.
And what did they reveal when you scan these people over time in relation to COVID infection? What does that reveal?
Regardless of whether people were 2 months after COVID, 6 months after COVID, a year after COVID, there were signs of increased inflammation and increased immune system activity in a number of different tissue sites, from the lungs to the heart to the GI tract, compared to people who had never had COVID.
Can you point the finger, though, at what's driving that sustained inflammation over time? Is that physical virus still there? Because one of the theories of long COVID is that people have some kind of reservoir of long-term infection that continues to tickle the immune system all over the place. Others have said it's almost as though the immune system just gets locked on like autopilot. So which do you think it is?
Yeah, so both of those things are certainly possible. And when we went into this, our framework for this was that the virus should come and go, and we shouldn't be able to find anything, beyond the first couple of weeks after a person's had COVID. So one of the really interesting things we were able to do in this study was, we saw all of the various tissues that lit up that had increased immune activity. And, you can't do brain biopsies on living people and heart biopsies. The risk is just too high. But one of the areas, as I mentioned, that lit up was the GI tract. And so, we were able to basically, in a subset of these volunteers, do a procedure called a flexible sigmoidoscopy, where a gastroenterologist goes in with a camera and takes little biopsies of the rectal wall, basically. And what we found in basically everybody who was biopsied, all these people had long COVID, was SARS-CoV-2 RNA that should not be there. And so that suggests to us that in at least one of these areas of inflammation that we're seeing, the GI tract, there is a relationship between that inflammation and pieces of this virus that are sticking around.
Interesting, isn't it? So is long COVID actually a persistent infection for some people? We'll have to wait and see. That was Michael Peluso from the University of California, San Francisco, and that study's just come out in Science Translational Medicine. To the world's most dangerous animal now, and that's the mosquito. These insects cause more deaths and disease than any other creature on the planet, and that's because they're the vector for a host of horrible diseases. But now, a new study has found that a pair of hormones work together to either drive or deactivate a mosquito's bloodlust. So maybe we can exploit them to stop ourselves being bitten. To find out more, we put in a call to the paper's author, Michael Strand, who's at the University of Georgia.
Mosquitoes blood feed on different types of animals, including humans, but after they blood feed, they lose interest in blood feeding again until they complete the formation of eggs, which is why mosquitoes actually blood feed at all. Blood by dry weight is almost entirely protein, and that protein is essential for egg formation in mosquitoes. Sugar is mainly used for energy for flight. It's kind of like taking an energy drink for a runner. And so males and females consume sugars mainly to extend their lifespan and to be able to fly.
Why not just feed on blood all the time, though? Because if it's got lots of protein but also some sugars, you could just live on blood all the time. So why don't they do that?
The general thinking is that blood feeding, as you can well imagine, is also quite risky for female mosquitoes because they can get killed in the process of trying to do it. So there is intrinsic risk associated with blood feeding that likely has mitigated why blood consumption is restricted to this female-specific activity as opposed to being the general source of nutrients for all activities. Feeding on sugars from flowers and the like is a much more low-risk occupation.
So it makes sense then to have this sort of switch. When I need to lay eggs, I go for blood. When I'm not doing that, which is a metabolically demanding activity, I go for the low-risk option. Those are the sugars.
That would be conventional wisdom, yes.
And have you got a handle now on how you think they're doing this?
We have a handle, or at least some new insights about what motivates females in terms of blood feeding. And maybe a convenient way of thinking about it is that just like human beings, we have periods of having a strong appetite. We're very hungry. And then after we consume a large meal, we're dissuaded from feeding further. In other words, we're satiated or we're full. Um, what we've identified is two hormones that control the motivation of having a strong appetite before blood feeding, and another set of hormones that convey a sense of satiation or fullness to mosquitoes after they blood feed.
What are the hormones? Where are they and how do they work? How did you show that this is what's going on?
These hormones are very short proteins, and we had antibodies which could bind to these peptides, and they would provide a marker so that we could see them in the mosquito's body. And we use these to be able to determine that one of the hormones is produced in a type of cell in the gut of the mosquito. And that hormone is called neuropeptide F, or NPF. And the other hormone is called rhyamide, and it's produced in neuronal cells that send projections to contact the gut. And then we could monitor their production and loss before a blood meal and after.
So which one does the blood meal turn on and which one does it turn off?
NPF is on before blood feeding and is very quickly turned off after blood feeding. And reciprocally, rhyamide is essentially off before blood feeding and is very strongly turned on after a female consumes a blood meal. So that means NPF functions as a promoting appetite, and rhyamide functions as suppressing appetite or giving a sense of being full. And we were able to show that because we could make these hormones and we could treat mosquitoes. We could eliminate the endogenous source of the hormone and we could replace it, kind of like therapy, and demonstrate that they had these functions experimentally.
Does this mean they're potentially a target then? If we could go after these, can we try to dissuade mosquitoes from feeding on us?
That would be a long-term aspirational goal, and there's precedent for using hormones in this way. A very important class of drugs that are used as weight loss control drugs today are administered to people, and they suppress appetite. And so one could imagine that molecules like this could be potentially delivered to mosquitoes and manipulate their behavior.
There's some evidence that mosquitoes being the most dangerous animal on the planet because they spread all kinds of diseases, things like malaria, things like the various viruses that we get from them, that some of these diseases, when the mosquitoes acquire them, change the behaviour of the mosquito and they make them bite more often. So is there any evidence that these diseases might be working through these hormone systems you've discovered and make the mosquitoes more likely to pass on the bugs they're carrying?
You're fully correct that pathogens mosquitoes acquire can manipulate the behavior of mosquitoes, and it's reasonable to speculate that one means of the way pathogens could manipulate mosquito behavior would be by modulating these kinds of hormones.
So you're going to have to come back in a year or two and tell us if that's the case.
Yeah, I would very much hope that we could do that.
I want to be on the paper if I'm right.
Absolutely. At minimum, in the acknowledgements.
We'll talk to you in a year or two then, Michael.
All righty.
Michael Strand from the University of Georgia. Fascinating study, just out in the journal PNAS.
The Naked Scientists podcast is produced in association with Spitfire, cost-effective voice, internet and IP engineering services for UK businesses. Find out how Spitfire can empower your company at spitfire.co.uk.
Music in the programme is sponsored by Epidemic Sound. Perfect music for audio and video productions.
You're listening to The Naked Scientists with me, Chris Smith. Still to come, we sink our teeth into the science of sinkholes. But first, to the Royal Society's Summer Science Exhibition, which has been taking place in the heart of London. It's a free event and no tickets required, and it's all about showcasing the best of British science. James Titko has been along.
You join me at the Royal Society's Summer Science Exhibition, showcasing some of the cutting-edge research from across the UK. I'm here in the main hall, eager to chat to some of the participating scientists to hear all about their projects. Hopefully, I'll be able to catch them as I'm wandering through, and I'll take you with me. Hi, nice to meet you. I'm James from the Naked Scientist podcast. What's your name?
I'm Ryan.
Could you sum up in a sentence for me the exhibit you brought to the exhibition today, please?
Yep, so we're engineering atom by atom. We're a collaboration between the University of Manchester, Leeds and Imperial College London, and we're showing how we can use ion implantation to create the materials of the future today.
Amazing. I mean, engineering atom by atom, so literally taking apart or putting together individual atoms.
That's right, yes. So we can take a single atom, we can charge it to create an ion, and then we can position that into another material with very high accuracy. And that allows us to create new applications such as quantum computers, topological insulators, or single photon emitters.
It's absolutely mind-blowing to me that engineering something so small can have ramifications for how we interact with our physical world.
Yeah, it's really exciting as well to be doing that, and it does change the material properties. So we can put in individual ions and then collections of ions that will change the material properties in ways that we can measure as well.
I know quantum computing's a highly hyped field.
Yeah.
What are the main challenges with achieving what it's going to be able to unlock for us?
So we're working on atom resolution, so 100,000 times smaller than the width of a human hair. And what we can do is we can also make these materials for our sources. So if we're implanting an ion, we need to have an alloy that will be stable and allow us to create that ion and then implant it into our material. So there's lots of scientists working on this project to allow us to improve the capabilities of this tool.
Well, it all sounds terrific. Thanks so much for chatting with me. Have a nice rest of the exhibition.
Thank you. You enjoy it.
Hi, I'm James from the Naked Scientist podcast. Can I get your name, please?
So I'm Dr. Ben Moe from the National Oceanography Centre in Southampton. So our planet is warming. At present, the ocean is absorbing 90% of this excess heat, this sort of anthropogenic heat that we and our ancestors actually, with CO2, have put into the atmosphere.
How on earth do you measure something like that 90% figure? Can you expand on the methodology there?
So since 2000, we have this program, this big international program called Argo, and we use these robots that basically measure the ocean for up to 5 years, and they measure the top 2,000 meters of the ocean. So we measure the temperature of it, its salinity, and we transmit that data back every 10 days. So sea surface temperatures are rising. So we've all heard about coral bleaching. So the Great Barrier Reef, for instance, is dying. But there are other consequences as well. As the oceans get warmer, it can hold less oxygen. 50% of the oxygen we breathe comes from the ocean. But also 30% of that excess CO2 is going to the ocean. That is then changing its pH. So that means it's harder and harder for these animals that build shells up around themselves to form. So there's a whole impacts for the whole food chain.
How are these findings that you've acquired through these techniques going to lead to things we can do, actions we can implement to improve that situation?
So what we need is large coupled ocean atmosphere, and link it to the ice as well, these big mathematical models to try and look at how it's going to change under different CO2 scenarios, carbon dioxide futures. We make the observations, we see how it's changing, we use those observations to benchmark or basically truth these big ocean models.
Terrific. Well, thank you so much for your time. Have a lovely rest of the exhibition.
Thank you very much.
Hi, I'm James from the Naked Scientist podcast. Can I get your name, please?
I'm Professor Gilly Forrester from the University of Sussex.
Tell me about Baby Boogie. What's the project?
So babies from birth have what we call writhing movements, and they're kind of like a belly dancer. They've got their arms and their legs going all at the same time. You see this kind of really smooth movement throughout the body, and it's relatively constant. There'll be bursts of it, and then it will calm down and bursts again. And we can see different kinds of features in this writhing movement. Clinicians have known for a long time that children who are in high-risk clinics, maybe they've had trauma at birth, that they can tell whether or not that repertoire looks healthy or poor. In talking with the clinicians and trying to better understand what they see in poor repertoire, and good repertoire, we've learned that the complexity and the variability of infant movements seems to associate with their cognitive development. So good high variability and complexity tends to associate with healthy cognitive development.
So we might see language development skills come a lot later in babies who would otherwise had those more fluid movements that you were talking about.
So not even necessarily later, but possibly disrupted if we see poor repertoire. And so the idea is that right now our screening for autism and other neurodevelopmental disorders tends to be quite late. We wait until we see a deficit or with language or social behavior. And so our diagnoses end up being 2 years, 3 years, 4 years, or even later. But what we think now we can do is find earlier risk markers that are visible and present within the motor behavior of babies from birth. The Baby Grow project is already revealing promising results. We're seeing that babies in the first 8 weeks of life are already showing differences in the high and low risk groups.
What are the benefits to those babies of understanding these developmental concerns as early as possible? How does that help you help them?
Oh, great question. The idea is that the earlier we can see risk factors, the earlier we can get in there and have new interventions. We don't have those interventions yet. They're things we need to develop, but we can develop motor interventions that help mitigate against some of those more severe language and social disorders that might happen otherwise.
Well, good luck with it. Thank you so much for your time. It's been a pleasure.
Thank you so much.
Big thank you to everyone that's given me some of their time. Signing off from the Summer Science Exhibition here at the Royal Society. Get down here if you get the chance.
Thanks, James. James Titko at the Royal Society's Summer Science Exhibition. That runs until the 7th of July, and it's free if you want to go and take a look. Now, you might have seen recent footage of a sinkhole appearing on a large football field in Illinois in America. The hole, which is 100 feet wide and 50 feet deep, swallowed the floodlights, benches and even the football pitch's turf. But what exactly is a sinkhole? Here's Vanessa Banks from the British Geological Survey.
A depression in the ground that doesn't have a surface drainage outlet, and therefore this depression is draining only from its base. The depression in the ground may have formed because of the lowering of the ground surface, because at that point there are soluble rocks. And a soluble rock is one that dissolves a bit like sugar in water when you pour slightly acidic water over it for prolonged periods of time. And as the hollow forms, then more and more water is focused to that point, and the dissolution extends to greater depth, and then we have something that we might call a dissolution sinkhole.
Vanessa Banks from the British Geological Survey. So can anything be done to prevent these sinkholes from swallowing up the ground? And might they actually have a practical purpose in some cases? We put in a call to civil engineer and science populariser Grady Hillhouse, who's best known for his YouTube channel, Practical Engineering.
There are lots of areas across the world that have the specific geology that that's conducive to sinkholes, and there's really not much you can do to stop the problem, right? It's very insidious. It's wide scale, so in that way, it's kind of threatening. But also, if you zoom out and look, we don't see that many sinkholes that cause real problems in the world, and so they're kind of rare, but also, it's a widespread problem.
So dramatic when it happens, but thankfully fairly infrequent at the moment. When it does happen, is there anything from an engineering point of view we can do that either we know will make the problem worse, so we know what to avoid doing, or if we build buildings, build roads, put in infrastructure in a certain way, even if there is a sinkhole in the vicinity, we're a bit more resilient?
Yeah, definitely. The first step of that is just identifying the hazard. And so a big part of engineering in places that experience sinkholes is just characterizing the hazard, right? And usually that involves drilling boreholes to take a look at the subsurface, see if there are caverns, see if there's limestone or gypsum or salt or human-made things like mines that could pose a hazard to a structure. And then once you understand that hazard, then you can make a decision about what to do about it. Maybe that means, relocating a structure from where you originally intended to put it. Or it could also mean building stronger foundations that, can withstand a little bit of movement, a little bit of settlement, or even bridge a gap that might form over time.
So if we know there's a risk in a certain area and we have some plans to do some building there, is there anything we can do to almost put sensors in or anything like that so we can tell how fast things are moving, when things are on the move, and therefore when to watch out?
Yes and no. There's definitely opportunities to instrument buildings and structures, and we do it all the time, right? To keep track of how a structure is performing over time, whether it be movement or expansion or contraction or cracks or, how groundwater is flowing. So we do that, not just for sinkholes, but for all kinds of things. But sinkholes are a little bit different in that a lot of times it's a very, very slow process that culminates in this really dramatic collapse. And so it's much harder to kind of predict that very last, the worst part of it.
I suppose one of the issues here is that most big cities sprung up because there was an abundance of mineral resources or something else people could get from the vicinity where that city grew from. And often that was stuff underground, which means most big cities, certainly from days gone by, were on top of resources that people mined out. So we've kind of made a rod to beat our own back, haven't we? Because we've got a city there because it was in an area that was plentiful for something we wanted. But the legacy of that is we're now building on land that we've turned into a honeycomb.
Yeah, I definitely think that's the case in some places. There are also human-made causes of sinkholes, like, for example, drainage and sewer pipes can get a crack and start eroding soil into the inside. And so oftentimes you'll see a sinkhole that opens up in the middle of a road. That has nothing to do with geology at all, right? It's all about a failure of a sewer or water pipe that's eroding the soil and moving it away from the subsurface.
Is anyone trying to do anything practical with sinkholes? So if we know that one is coming or one has happened in an area, it sort of saves us from making a big hole in the ground, it strikes me. So is there anything that people are saying, well, this could actually be useful?
That's a good question. There are definitely examples where natural drainage will flow into a sinkhole, and that can be useful to recharge an aquifer that may be used as a water supply. I'm trying to remember the Arecibo telescope in Puerto Rico. That was formed in a big basin, and I can't remember if they would call that a sinkhole or not, but that was an example of, using the natural topography to the advantage of creating a big radio reflector.
Thanks very much to Grady Hillhouse from Practical Engineering there. Well, now it's time for Question of the Week, and James has been taking on this inquiry from listener Donald.
Why don't plants get cancer from UV exposure like humans do? Thanks, Donald. You're right to point out that humans can get skin cancer from UV radiation, the most dangerous of which is melanoma. This is when UV light damages DNA in melanin-producing cells in the deepest layers of our skin. Over time, enough DNA damage can cause cells to grow out of control and lead to cancer. So why does something similar not happen in plants? Well, here to help me answer that question is Gareth Jenkins, Professor of Plant, Cell and Molecular Biology at the University of Glasgow.
The answer is that plants have evolved very effective ways of protecting themselves from the harmful ultraviolet radiation in sunlight that causes sunburn and melanoma in humans. Although we humans can shelter from bright sunlight, exposure to UV radiation is an unavoidable hazard for plants. They need to be exposed to sunlight for photosynthesis, which fuels their growth. Plants avoid damage by sunlight in three main ways. Firstly, they reflect it by having a waxy coating and hairs on the surface of leaves, but this reduces light exposure in general and doesn't prevent UV exposure specifically. Second, plants actually make their own sunscreen and deposit it in the outer tissues. These sunscreen molecules work in the same way as our sunscreen by selectively absorbing UV wavelengths.
Well, that's a whole lot more convenient than applying sunscreen out of a bottle. And now for that other solution.
Thirdly, plants have effective mechanisms to repair any damage to their cells caused by UV exposure. An important feature of all these protective mechanisms is that they're stimulated when plants are exposed to UV radiation. Even very low levels of UV, such as found on a dull winter's day in the UK, are sufficient to stimulate production of the sunscreen and UV repair mechanisms. This ensures that protection is already in place when plants are subsequently exposed to high levels of UV in bright sunlight. Remarkably, plants can detect the presence of UV using a photoreceptor protein, and this causes genes to be activated that enable production of the sunscreen and damage repair mechanisms. So plants are able to avoid damage in sunlight through genetic processes stimulated by their ability to sense and respond to UV light.
What a stimulating question you've asked there, Donald, and what a comprehensive and yet concise answer from Professor Gareth Jenkins from the University of Glasgow. Thanks so much to you both. Next time, I'll be answering this question from listener Phil. He says, according to particle physics, at the smallest scales, empty space isn't empty. Instead, it's a hectic place with subatomic particles appearing and disappearing. So what replaces the space that the particles occupied? Is this a void?
And if you think you know the answer, or you have a question you'd like us to delve into, do drop us a line. It's chris@thenakedscientist.com. You can also join us on our forum. That's at nakedscientist.com/forum. Well, that's where we're going to have to park it for this week. Do join us on Tuesday, though, when Titans of Science continues with our British Formula One engineer, Dan Fallows. He works for Aston Martin and makes F1 cars go incredibly quickly. Just like the time on this programme when you're listening. And look, just before you go, if you would like to support us, we do rely very heavily on donations to keep the programme going. And if you appreciate what we do for you each week, then do please consider heading over to nakedscientist.com/donate, and you can very safely and very securely make us a contribution to help us with the production, creation, hosting and distribution of this programme. We really would appreciate it. nakedscientist.com/donate. The Naked Scientist comes to you from the University of Cambridge's Institute of Continuing Education. It's supported by Rolls-Royce. I'm Chris Smith. Thank you for listening. And until next time, goodbye.